Thank you all for being here. I'm Katja, and I'm very excited to take you on a quick tour through Yatir Forest, where pines are trying to keep their cool under growing heat stress and where their future carbon storage is on the line. Trees are often planted for climate mitigation, and because they help to cool the planet and they capture carbon. And recent just became louder, right? And recent research has shown just how valuable trees can be for absorbing carbon and for supporting ecosystems in many ways. And although all of this research here and what we see sounds very promising and very clear, how do we actually tree stem growth, which reflects a slow, there's somehow an echo, no? Do I hear it? Okay. I think it's from the Zoom. Maybe I put this on. Maybe like this, sorry. What was I saying? Okay, so tracking tree stem growth reflects a slow long-term development. In contrast, eddy covariance flux towers, they capture fast, short-term responses of forest functioning by measuring carbon and water fluxes in real time, such as, for example, during a drought or a heat wave. And although forest monitoring can give us a really good snapshot how of the forest or an ecosystem's present state, it cannot predict how a forest will change in the future. So one question that remains, I still have an echo here from the computer, but it's okay. So one big question that remains is dryland forests. They're widely proposed for afforestation. How will they actually be doing in the climate that we're heading towards? Because dryland forests, they're already adapted to drought, to heat and aridity, but we don't know in a future climate, will the climate push them beyond their threshold, beyond a certain limit, or are they adapted enough to just continue doing what they do, or might they even benefit somehow from the future climate? So what we want to find out in my research, how will future drought and heat reshape dryland forests structurally and functionally under rising atmospheric CO2? And what happens to the carbon stored inside those trees over time? Because if trees might die from heat, drought stress, where does what happens to the carbon that is stored within these trees? Where might it go, or how much carbon could be lost? So, welcome. So one of the forests that I study is Yatir Forest, that most of you probably know. It was planted in the 1960s, and it is, maybe you can correct me, it's the driest, or one of the driest planted pine forests in the world. And it was once seen as a success story because despite being planted in desert, it was actually doing quite well. But nowadays, or like in recent times, it's been showing more and more signs of decline because there aren't really any new trees establishing. The woody cover is becoming patchier, and water is increasingly limited. But because Yatir has been monitored closely for decades, it gives us it's an ideal case to study how a forest has already been responding to ongoing climate change. But to not only look at the past, but also to explore Yatir's future, I use the ecosystem demography model ED2. And this is a process-based simulation model that represents different plant functional types, and it simulates their physiology, their growth, their competition and mortality in response to environmental conditions. And the model specifically tracks water, energy and carbon exchange between vegetation, the vegetation, soil and the atmosphere which allows us to track the forest structure by, for example, looking at the canopy and the biomass, as well as its functioning by, for example, photosynthesis and water use. The model is parameterized with local species traits and local climate data, and then we compared it to observations to make sure that the model represents dynamics that are that correspond to your observations at Yatir. So, for example, two things that are important to us and that the model can simulate are, for example, how much water is available in the soil after it rains, and how much carbon is absorbed or released by the forest in terms of net ecosystem productivity, as you see here. And so we compare the model simulations with observations, and then we change the parameters within like a known range for us until the lines closely match. And we also did this for LAI, leaf area index, for evapotranspiration, and for growth. And we adjusted the parameters as much as we could until we got like realistic fits. And with these final settings that we got from our calibration and validation process, we tested 3 different climate scenarios. One is basically like the current climate recycled from 2015 until, all of them are from 2015 until 2100. Then we chose an intermediate climate projection, the SSP 370 scenario, and we also chose the worst climate scenario, the SSP 585. And with these different, and for each of them we modeled 5 different models within these climate scenarios. And with these different conditions, we modeled how the forest evolves structurally and functionally, and what the main drivers are for the observed dynamics, as well as for the differences between the environmental conditions. And our simulations showed the following predictions. All of these 4 figures here have basically the same organization, where we have time on the x-axis, we have ecosystem response on the y-axis, and the colors are the different climate scenarios. The orange ready one is the worst one. The yellow-orange one, I hope there's no colorblind people, it might look different to you, is the intermediate one, and blue is the current scenario. And basically what we saw is that the forest, for all of these structural variables that we looked at, the forest keeps growing structurally until the end of the century. First steeper, and then it kind of plateaus. And in the case of ATB growth rate and leaf area index, at some point it doesn't plateau, but it declines. What was interesting about this increase in biomass accumulation, that it mostly stems from the originally planted trees, because there's no demographic renewal in our simulations. So all of the trees that we saw at the end of the simulations are mostly like the older, taller version of the young trees that we started with, the ones that are left, because some of them have already died. And when we looked at the drivers for the growth that we saw, green is positive drivers, and the salmon color is negative drivers from -1 to 1. And we saw that the most positive drivers for almost everything was CO2 fertilization. And this kind of corresponds, in early mid-century, late century drivers. And that CO2 was at 5 minutes. Yeah. So CO2 was the main driver in the early beginning, as we also saw in the growth slide before. But then towards the end of the century, they're mostly driven by water availability. As I said at the beginning, trees are often planted for how much carbon they can hold. So we also looked at NEP, net ecosystem productivity, and how much the forest absorbed versus released. And in the beginning, so below this dashed line, it's a carbon source, so it releases, and above it's a carbon sink. In the beginning, it's mostly a strong carbon sink, not source, sorry. A strong carbon sink. It really takes in a lot of carbon, but then towards the mid-century, it flips more and more, and then towards the end of the century, it's becoming mostly carbon source. And in terms of water use efficiency, shows a very similar dynamic with an increase and then a decline in function over time. The drivers for this are very similar to the structure, where carbon fertilization plays a really important role in enhancing this growth in the beginning, but forest function was from the beginning until end also strongly controlled by water availability. So, to summarize, we saw that the forest could grow, could continue growing structurally thanks to its drought adaptation. We also saw, despite its functional performance starting to decline, we also saw that CO2 can help to enhance this growth, but only under moderate conditions, because once the heat and drought stress intensified, the forest function declined, and the forest's ability to store carbon and to function broke down. And so what we saw is that forest structure can mask functional decline, and also that monitoring structure alone is not enough to have a complete picture of how the forest is doing. And what we additionally to this research want to find out is what actually makes a forest resilient in the long term, or how can a forest be managed to be sustainable in the long term. Because only because a forest is resilient today does not guarantee persistence tomorrow. That's it. Thank you for listening, and special thanks to these people, and I look forward to your questions.
Does the model capture scale-dependent feedbacks that may lead to emergence of patterns, special patterns? Because this is a population-level stress adaptation mechanism that can affect the resilience of the system.
So what exactly do you mean with scale-dependent? Yeah. So the model can definitely capture this to some extent, because the model is a cohort-based model. So it can look, in the beginning at least, it can look from the individual to like the whole ecosystem. So we can really like zoom in and zoom out what are the interactions between at different scales. But for computational efficiency, these individuals are more and more summarized into cohorts of similar sized trees, so we lose a little bit of the individual information, but technically it is possible to look at these effects. This is a really good question. Thank you. It was about mortality in the model. The model has a lot of different types of mortalities that it takes into account. So there's age-dependent mortality, and we don't really know how long the pines in Yatir are going to live, but there's a higher probability at the higher age, and I don't think they can live longer than, I'm not really sure what the number is, but it's like probably not even 150 years or something. It's definitely less. Apart from the age-dependent mortality, there's a drought-dependent mortality, then there's a random mortality that takes into account that sometimes there's pests of different kinds. And also there's one that stems from competition. So if there's, this is not, it's not really so important in this case because there's no new trees establishing that could steal a lot of the water away, but it's still important because if water becomes limited, then there is more competition for the limited resource.
Very important work. Thank you, Katja. So next speaker today is Oren from Tel Aviv University. Yes. So we're moving from forest to shrubland. Okay. Meantime, I want you to give a good clap to Asha. Thank you, Asha, for helping. And Israel, please.
Thank you, Tamil. Hello, everybody. So today I'm going to tell you about my work in the Mediterranean shrubland and a little bit of background to my work. So climatic models predict an increase in the prevalence of extreme weather events. They include higher maximal temperatures, decreases in annual rainfall, and increase in heavy rainfall events, and longer dry spells in between rainfalls. All of which will decrease the soil water availability, which combined with the disruption in weather patterns, in rainfall patterns, should affect the plant functioning and growth, with further implications to ecosystem stability and functioning. So in light of these predicted reductions in rainfall and disruption of climatic patterns, I wanted to know whether Mediterranean perennial plants will be affected by these changes, and if yes, would it be because of the reduction in the precipitation or because of the disruption of the rainfall frequency, or perhaps they would impact equally? So to do this, I worked in Mata, a long-term ecological research station, which is a Mediterranean shrubland located in the Judean hills near Beit Shemesh. It is located 640 meters above sea level with an annual precipitation of 540 millimeters. And in Mata, a long-term experiment has been established in 2019 in which the effects of reductions in precipitation and changes in rainfall frequency are being tested. So to do this, 4 different types of plots have been established. The first plot is a control plot in which there is no manipulation of rainfall, quantity or frequency. Then there is a second type of plot to test the effect of rainfall reduction, in which we have gutters that intercept 66% of the natural rainfall. So we reduce the rainfall quantity but keep the natural frequency. That's the natural frequency treatment. And a third type of plot, we completely exclude water, so then we can control also the frequency. We exclude the rain and then we add it through sprinklers. In either high frequency treatment with 9 small rain events, or with the low frequency drought treatment where you have 3 large and rainfall events. But again, the total annual or the seasonal amount of rainfall is 66 less than the mean annual precipitation. In Mata, there are plenty of species, and I decided to work on 4 perennial ones, 2 geophytes and 2 shrub species. The 2 geophytes were Asphodelus ramosus, Tsirit Gdola in Hebrew, and Cyclamen persicum, Rekefet Metsuya, and 2 shrubs, Pistacia lentiscus, Elata mastic, and Salvia fruticosa, Marva meshuleshet. All the 4 species can be found in Mata. They are very common to Mata. So I conducted 2 campaigns, a winter campaign in which the conditions are ideal for plant functioning, and a second campaign in the summer where conditions are the most demanding. Of course, in the summer, geophytes were already dormant, so for the summer campaign, there are only the shrubs. Also, I'd like to mention that 2025, the year I conducted my work, was extremely dry with just above 50% precipitation compared to the mean annual precipitation. So if I want to understand whether the treatments in Mata actually affects the plants, I can look at different variables. Does it look good? Variables. Just a second. Okay, so present it like this. Different variables that if I see a response compared to the control, I can conclude that there is some stress going on the plants. And these are variables that I looked, and I'm going to walk you through them now. I looked at functional and water relation variables such as photosynthesis and transpiration, which if there is some drought stress, I would suspect the gas exchange to reduce. And then the water use efficiency, which is the ratio between the amount of water lost per the amount of carbon CO2 fixed by the leaf, it should increase. Then the relative water content should reduce when water is limited. And I looked at the damage indices such as chlorophyll content index and the fluorescence of photosystem 2, which is supposed to decrease. And ion leakage is supposed to increase where cell membrane stability is deteriorates. Also, limited water availability should cause some morphological changes which mean to reduce the water loss, such as reduction in leaf surface area, increase in leaf sclerophyll, which is the leaf mass area. Leaves become more sclerophilous and thick. And then the biomass proportion between the leaf and the woody tissue, it's supposed to decrease where plants become less leafy. And then the integrative response is growth. And in growth, I present you now the branch elongation rate as a measure of growth. So when I look at photosynthesis in the geophytes, there was a photosynthesis responded to the ambient conditions, either to light or to VPD, but also to the treatments. There was a significant difference between treatments, but with a tendency to increase with a high and low frequency compared to the control. And when I dissect whether it was because of the quantity or the frequency, so only the frequency significantly affected the photosynthesis in these 2 species. In contrast, in the shrubs, there was a response to the ambient conditions, but there was no difference between treatments in photosynthesis. And a similar view I found in the transpiration, although transpiration responded less to the ambient conditions. But when combined together with the water use efficiency, so there was no difference in the geophytes, Pistacia lentiscus, and only in salvia, in the natural frequency treatment, there was a significant increase in water use efficiency, suggesting something was going on with salvia. Damage indexes, such as the fluorescence of photosystem 2, I found no difference in either of the species between treatments, and also the chlorophyll content index. It didn't differ between treatments at all. Looking at leaf surface area in the geophytes, so in the high frequency treatment, either through the leaf surface area or the leaf mass area, it seems that the geophytes benefited from the high frequency treatment if I compare it to the control. But in the shrubs, I found no such difference between treatments. Looking at the leaf weight ratio, here it's an analysis of the data from the winter and the summer combined. So in Pistacia lentiscus, there was no difference between seasons and treatments. The leafiness was the same. In Salvia, there was a difference mostly between seasons and less so between treatments. There was a slight treatment effect, and then again it was manifested through the frequency. Looking at the branch elongation rate, again, no difference between treatments, just between seasons. So it was dictated by the phenology, but not the water availability. Two last additional kind of damaging indices, only in the shrubs. There was no difference between treatment, the ion leakage, and in the water content of the leaves. But I didn't compare it indirectly, but if you look at the difference between Pistacia and Salvia, Salvia suffers more overall from the conditions in Mata. So as a short summary, in the many variables that I examined, I found no difference compared to the control. And if there was a difference, it was in the opposite direction to what I would have expected if there was a drought stress experienced by the plants, which suggests maybe some acclimation. So, were the plants stressed? Probably not the shrubs, but Salvia fruticosa was not well off as Pistacia was. Regarding the geophytes, there was some sensitivity to the treatments. In this very dry year, the control performed better than the high and low frequency treatments, which suggests maybe some sort of acclimation. Regarding the second question, was it were the responses because of the frequency or the quantity? So I found that it was because of the frequency, the alterations in the frequency, and not the extreme reduction in precipitation. And then it leads to future questions of how do they do that. I suppose at the moment that the shrubs have access through extensive root systems to some water pools that disconnect them from our treatments and from the annual pattern. Either directly they reach some deep soil layers or take up water from rocks, or maybe it's being done through mycorrhizal relationship. Also, that's probably through anatomical branch and leaf anatomical changes that can accommodate the demand due to our hydraulic limitations. With the geophytes, probably it's the tuber that buffers extreme years, as it was this year, and probably also through changes in the biomass allocated to vegetative and reproductive tissues, probably in favor of the vegetative in harsh years. If I have time, my next steps would be to analyze more data that I still have, such as the summer gas exchange campaign, quantify above ground biomass as an additional measure of growth, quantify treatment effects on non-structural carbohydrates, because it is the drought also supposed to affect this variable. And I was talking about quantity of water all the time, but not about soil matter, which is important. It is for the roots to actually pull water from the soil or from the water pools. So we recently installed soil water potential sensors, so we will soon have a lot of data to work with. I would like to extend measurements to a third shrub, which is very common in Mata, in the Mediterranean region, Sarcopaterium spinosum, and analyze potential anatomical features in the shrubs, and also to understand the water sources from which the shrubs at least take up water, which probably can be done through stable isotope studies, and also to look into potential mycorrhizal relationship. So this could be a collaboration, future collaboration. And I'd like to thank you for your attention.
Thank you, Time for questions?
Very interesting study. I was wondering, are you planning on repeating that over several years? Because from your finding, it sounds like these plants may be adapted to short-term droughts of a year or something, but what would happen over several years if this were to continue 3 years, 4 years, 5 years?
Monitoring can be done in the future, but here I entered an experiment that has been running for 6 years. So the plants were exposed in the treatments for 6 years of drought now. So I was expecting to find an accumulated response, even I didn't monitor it from the beginning. And yes, probably I will repeat it, at least some of the variables, but I don't know for how long.
And a small technical question, how deep is the soil? And were you able to see roots in the soil or in rocks? Yeah.
So in most places the soil is very shallow. And I didn't go into roots because it's a very dense shrubland, and at this point I didn't get into it. I would like, but it's just to identify the roots of our shrubs. It's going to be a major task that I should be focused on. So something for the future for sure, because through the above ground there's nothing going on there. So it's probably down below.
Thank you very much, Israel. Okay, moving on to Hemanth. Yeah, welcome. And you're talking about brassica crops. So, looking forward. Thank you.
Thank you. So I'm Hemanth. Today I'm going to talk about my master research project, which is the impact of reduced rainfall on the performances and pollination services of 3 Brassicaceae crops along with their wild relatives. Okay. So I like to begin with highlighting the issue which we are all aware of, which is the climate change. So one of the evident impact of climate change is rise in global temperature. So here in the graph we can see over past few decades we see an increase in the temperature, and it is tend to increase even more in the upcoming years as well. So one of the evident impact of rise in temperature is also alterations in the precipitation patterns. So this has resulted in reduction in rainfall in many regions. This trend is quite alarming to the regions which are already facing the water scarcity, such as the Middle East itself. And we can see here this, if this situation continues, this is going to become worse in the upcoming decades. So this water scarcity or this reduction in rainfall has a direct impact on the food production, which leads to food insecurity in the region and also in the global level as well. So here in this map we can see how the food insecurity going to become worse in the upcoming years. So in order to maintain food production and food security, it is also necessary to maintain pollination services. So nowadays in recent times we can see due to global change and land use intensification, the pollinator diversity are getting reduced. So these pollination services mainly depends on the floral volatiles or the overall plant emissions, which is also known as volatile organic compounds. So these volatile organic compounds helps plants attract pollinators and also to repel the herbivores. So in my experiment, I will be working on the Brassicaceae family, which are mainly cultivated to produce edible oils, vegetables, and also fodder for the cattle. These species are diverse in the Mediterranean regions. Some species are also domesticated locally in the Middle East as well. So these three species are my experimental species, which are Sinapis alba, Brassicana pus, and Raphanus raphanistrum, along with their wild relatives as well. So in this experiment, I had 2 study questions, and I will also explain the predictions that I had before conducting this experiment. So my first study question is, do domesticated crops and wild relatives respond similarly to the less rain? So I predict here wild will actually perform better than the domesticated, because wild already adapted to the local climatic conditions and also to the less rain, or also experienced drought stress already. But when we look at the domesticated, these species mainly depend on the humans for irrigation, for water, and also for the fertilizer. So I predict that domesticated will perform lesser compared to the wild. And my second study question is, do water changes affect floral traits and pollination services in domesticated and wild crops? So here I predict in domesticated, due to its animation is not working. So this is the aerial view of the experiment that I conducted. So as I said before, I had 3 species along with their wild counterparts, and I also had 5 levels of irrigation, which is 0.5, 1, 2, 4, and 8 liters per hour, and I had 6 replicates for each treatment. So in this experiment, I have measured physiological parameters and also have looked for floral emissions by using solid phase micro-extraction method, which is also known as PME, and I also conduct the pollinator observations. So now let's look at the results. So these are the physiological parameters that I measured. So here we can see in all the 3 species, we see the irrigation had an effect. So higher the irrigation, higher the transpiration rate in all the 3 species. But when we look at the Raphanus, here we can see the Raphanus wild actually perform better than the domesticated one towards the higher irrigation. We see the same results in stomatal conductance as well. And then if I have to look at the overall plant biomass, even the plant biomass treatment had an effect even on the plant biomass as well in all the 3 species and along with their wild relatives. So now let's look at, what is this? Sorry. So now let's look at the VOCs, the flower emissions, the volatile organic compounds that what and all the analyze I did. So this is the PCA of Sinapis alba. This is showing a clear difference of VOC emissions between the wild and the domesticated species. But when I look at the treatment level, the treatment had no effect on the VOC emissions in both the wild and the domesticated. And we see the same results or the same trend in the Brassicana pus species as well. So here also there was treatment had no significant effect on the production of VOCs. So the same follows in Raphanus as well. Then I wanted to group these compounds into specific classes based on their biochemical pathway. So here you can see the classes that I grouped into. So here also when I look based on the class level, here also I saw a significant difference between wild and the domesticated species. But you can see the irrigation in the x-axis, the irrigation had no effect on the classes as well. And the same follows in the Brassica napus and also in the Raphanus. So later, I also wanted to check like if the treatment had any effect on the pollinator visitations, but unfortunately I didn't find the effect of treatment on pollinator visitation. So I wanted to check if the number of flowers had anything to do with the attraction of pollinators. So here I saw the number of flowers had an effect on attracting more number of pollinators, which means more number of flowers attracted more number of pollinators. So what's interesting was the Sinapis wild actually attracted more number of pollinators compared to the domesticated one. And here also we see the same trend. The Brassica napus wild attracted more number of pollinators compared to the domesticated one. And then Raphanus also we see the same thing, but here it's quite stronger because even though the Raphanus wild has fewer number of flowers, still they manage to attract more number of pollinators compared to the domesticated one. So, I like to conclude by saying the irrigation significantly altered the physiology of the plant, but when we look at the Raphanus, we saw that Raphanus wild actually performed better than the domesticated one. This kind of proves my first study question. And second thing, we saw that irrigation had no effect on the floral VOCs. But when we look at the crop or the variety level, the wild and domesticated VOCs significantly varied. Sorry. I don't know what's No. It's next slide, but it's not showing the next slide. Yeah, it's something technical, guys. Give me. Good. Okay, just read the slide. Yeah. So when we look at the pollinator observation, so when we look at the results of pollinator visitation, so here we saw more flowers attracted more pollinators in few species, but what same? It's not coming again. Okay, I will say. So what was interesting was in all the three species and its wild relatives, the domesticated had fewer number of pollinators, but the wild species has more number of pollinators. So this proves the second study question that my experiment had. And there's no PPT. Okay, so I like to thank you all for listening to my presentation. And sorry for the disturbance in between. And I also thank my supervisor for guiding me, and I thank my friends for helping me in my experiment, and I also thank the SAGE Center for funding this experiment. Thanks, guys.
Thank you for bringing this. We still have time for one question.
Thank you very much. I was curious for the visitation, if it was just a manual observation or could you use cameras? And secondly, the practical implication, do you think breeders should then take traits from the wild type and integrate it into the commercial variety? Would that be the follow-up step based on your research?
it was manually done, and this was repeated 3 times to make sure, there will be errors for sure while doing the pollinator observation, but I made sure that by making 3 repetitions of all the 3 different timelines that I took, maybe I was confident with the result that what I found. And I just forgot, what was your second question about? Why? Can you repeat again? Oh, what can we do with kind of results? So maybe like what I understand from this experiment is like maybe the domestication has lost some traits along the way. This is the reason why the wild is still attracting more number of pollinators compared to the domesticated one. So maybe like in future it will be important to find what were these traits that we lost during this domestication process. Yeah.
Thank you, Hemanth. We apologize for these difficulties, but we are trying to solve them. Okay, so next speaker is Dr. Oded Cohen, all the way from Golan Heights, and I hope this one will go well. So let's try. This is for the right hand, and this is for the left.
Thank you, Tamir, Ash, and thank you for all the organizers. Hello, everyone. Many times we push our children toward the mainstream, believing that following the common route is the safest way to succeed. Over the next few minutes, we will see that sometimes stepping away from the mainstream and choosing a unique, unconventional path can also lead to remarkable success. This study dealing with the spread of the Sandara gum tree, Tetraclinis Articulata, is the results of a collaboration between Gil Osem, Efrat Shefer, myself with my research assistants, Nitzan Bar Shmuel, Gil Weber, Aviv Guy, and Tamir of Yoz. And was founded by the Jewish National Fund. Mediterranean plant communities are relatively resistant to biological invasions. The climate is arid, and the native plant community creates shading, while competition for water and light makes it difficult for invasive plants to establish and spread. It is often assumed that invasive plants are mainly defined by their high competitive ability. But that is only the half of the story. Many invaders actually avoid direct competition and specialize in disturbed habitats where exciting vegetation is thinned out and competition for resources is greatly reduced. Early successional colonizers are a common trait of many woody invaders in drylands. They are typically characterized by high light demand, rapid growth, short juvenility, and prolific seed production. So we often find these species thriving along roadsides or in disturbed wet habitats where they benefit from improved water and light availability and reduced competition from established vegetation. Tetraclinis articulata is a conifer tree native to the southwestern Mediterranean. Thank you, Ash. It was introduced to Israel afforestation and has been planted across a wide range of habitat, especially in semi-arid regions. In its native range, the species typically occurs in areas receiving about 400 or 1600 millimeter of annual rainfall, similar to part of Israel. At the same time, natural population in its home range have declined, probably due to the human activity, and the species is considered endangered there. At first glance, it is hard to think of Tetraclinis as an invasive species because it grows slowly and has long juvenile phase. On the other hand, the species produces large quantity of seeds, is capable of vegetative reproduction, and the new environmental conditions in encounters in Israel closely match its ecological niche in the native range. Reports of this species as invasive elsewhere in the world are very limited. Initial observations of young trees spreading beyond the planted stands were reported in Ramat HaNadiv in the north of Israel, something like near Haifa. Okay? It was not clear whether this was just a local phenomenon or an early warning of wider invasion. The main goal of our study was to assess the naturalization and invasion status and invasion potential of Tetraclinis articulata in the eastern Mediterranean. Specifically, we aim to quantify the abundance of Tetraclinis saplings within plantations, describing naturalization, and beyond plantations margins, describing invasion, and to identify the silvicultural and environmental variables that influence the extent of this naturalization and invasion. In this presentation, I will focus only on the invasion process outside the plantations. We conducted a national survey across 46 planted stands of Tetraclinis, all over 30 years old. These plantations were distributed across two contrasting climate zones, Mediterranean and semi-arid. To assess the invasion beyond plantation margins, we established systematic transects at 4 different distances from the plantation borders. We sampled a total of 1,588 transects extending from 10 meters within the plantation edge to 16 meters outside. Everything is okay? Allowing us to quantify the spatial distribution of naturally regenerated sapling of Tetraclinis articulata. In each transect, we recorded key environmental variables such as hard bedrock, rock and stone cover, slope, aspect, vegetation type, and climate type, as well as forest variables including stand maturity, planted tree density, and tree height. Our data show that sapling density declines sharply with distance from the plantations, and this pattern is strongly shaped by silvicultural and environmental factors. In the Mediterranean zone, see graph 1, Tetraclinis saplings reach very high densities close to plantations borders, with more than 1,000 saplings per hectare within the first 10 meters, but their density drops steeply beyond 30 meters. In contrast, regeneration in the semi-arid in the semi-arid zone, in much lower, indicating that wetter conditions near Mediterranean plantations are crucial for seedling establishment. Plantation maturity also play an important role. Sapling density are significantly higher in mature than in young plantation. This is figure B. And the highest density occur near taller trees, see figure C. And we observe a strong preference for higher sapling densities on north-facing slopes. As seen in the graphs, Tetraclinis shows a clear preference for tall vegetation formations. In Mediterranean regions, it established mainly in forest, but shifts to woodland in semi-arid. This preference for forest-type vegetation is consistent regardless of distance from plantations. Finally, we found that sapling densities were particularly high in rocky areas that were not grassed. See figure A. Grazing interacted with sapling size. Densities of large saplings were much higher in ungrassed plots. than in grass plots. In the following slides, I will present some field examples of how Tetraclinis invades beyond plantation boundaries. Here we see Tetraclinis seedlings establishing within pine forest. In this case, we see Tetraclinis saplings actually colonizing the scrubland in the Park Canada, further illustrating their ability to invade diverse habitats beyond plantation boundaries. And here we see Tetraclinis saplings establishing under the canopy of eucalyptus forest in Tsipori forest, showing that the species can successfully colonize shaded forest conditions. In this photo, you can see mature Tetraclinis trees with scattered saplings establishing around them. In recent years, there has been a growing interest in invasive plants that are able to establish beneath forest canopy. These species are often called superinvaders. Most research on this unusual archetype comes from temperate and tropical regions, where water is not as limiting as in dryland. For the first time, we see that this pattern of superinvader is now documented in Mediterranean climate as well. The superinvader archetype differ from the common archetype. It colonizes habitats at later successional stages, is relatively shade tolerant, grows and matures slowly, depend on more strongly on the specific conditions in its germination niche, and has more conservative carbon use strategy. Until now, afforestation efforts have focused mainly on importing early successional tree species that grow and mature quickly. In recent years, however, there is increasing interest in drought-tolerant trees with more conservative energy carbon use, and with it, a growing concern about introducing species that fit the superinvader archetype. These risks need to be carefully considered by decision makers. At the local scale, cutting trees is always painful, but bold action will be needed to reduce seed production of these invasive species. Such efforts should start in the Mediterranean climate, with particular focus on plantations adjacent to natural woodland. One of my favorite lines in Hebrew music, written by Micha Shitrit, deals with the question of choosing a life strategy, to be one of a million or one in a million. My personal takeaway from the Tetraclinis study is to let go my own ideas about the right way to succeed and to give my doubters the freedom to find their own way to success, which is not always easy for me as a father, but sometimes it works. Thank you.
Thank you, Oded. Questions to Oded? Thanks.
Thank you for the talk. You described two different strategies for establishing yourself as an invasive species, and what I would like, if you can tell me, to know is it because it is different from the rest? Like, is it because the species has chosen a slightly different way of establishing itself, or is it because it is just better? So, in another word, if all plants chose this, the second option, the long, how do you call it? The slower approach, would it still be better, or is it because it is special?
I think that there is two factors in the Mediterranean climate. that limiting the invasion. One is the water. Israel is in Dryland. And the second is the shading of the natural vegetation. So almost all of the invasive plants that I think about, they establish in disturbed habitats where the resources are, the availability of resources are increased, okay? But we need to think that there's all in the history we introduce species that are fast-growing trees, and not and they can grow fast and spread in the landscape. But the demand of species that are adapted to the dryland is a new phenomenon. And I think that in the future, we see that this archetype is more invasive than we thought. So we introduced thousands of species with a lot of strategies. And this is a new strategy that we introduced in the late 10 years. Yeah?
Okay, thank you, Oded. I'm sorry, we don't have time. Thanks. So next speaker is Amnon Kochavi, going down below ground.
Okay, good afternoon, everybody. My name is Amnon Kochavi. I'm from the Volcani Institute, and I'm going to talk today about the root system, how root system can assist to adapt crops to climate changes, and specifically about watermelon. So, a quick overview, although we already talked about it today. So, as been discussed before, over time, watermelon breeding has focused mainly on improving fruit yield and quality. The region of watermelon is from the arid regions in Africa and been shifted through Asia to Europe and to America, and over the process of domestication, many drought and other stress-related traits have been lost. As we know today, modern watermelon is it's water-demanding crop, and it's very sensitive to water limitations. And as you can see here, it's been predicted to have over 30% yield loss in the year 2050, according to the current models of climate change. In 2020, extreme weather in the US have been to $500 million loss. $500,000 cost loss of the watermelon crop in the regions in the southern areas of the US mainly. And even here in Israel, this year, as we know, is not the last year, 2025, wasn't that good year. And many farmers have decided not to grow watermelon this summer because there's water limitation, mainly in the northern areas when the soil is in the spring. And they have a bad winter, so they didn't plant watermelon and go to other crops instead. So what we try to do is we try to take the wild watermelon, Citrus colocynthis, Avatiach pakoa in Hebrew. It's very common here in the desert. You can find it in many areas around here, and in other countries in very arid conditions. And we want to see if we can find traits that we can take into the domesticated watermelon, the Citrus lanatus, and improve its drought tolerance without having yield loss. So what we do, we take a physiological measurement. We grow our plants inside this tube, this 1 meter high tubes, 8 diameter, full of sand. We have two types. We have the Citrus lanatus, the domesticated. The SB means sugar babies, some sort of watermelon. It's very easy to breed, so we work with that. And the wild, the Citrus colocynthis aro, it's because it's tagged, it's been found in Chatzeva area by people from Neve Yaar. So we grow them in these tubes in order to see how the root system responds in the different depths. We measure the soil water content in 4 different depths in this tube. We make small holes. We took TDR and take measurement over the experiment. And we took the geological measurement above the ground level. We work with the LICO 6800, LICO 6800, the porometer, the LICO 600. And also we took leaf samples for osmolyte measurements and for sections. The root system, in the end of the experiment, the root system was analyzed according to the different depths. We decided to divide it into 4 sections, from 1 meter until 75, 75 to 50, 50 to 25, 25 0. So here for the results, you can see here, this is the soil water content over the experiment. The experiment took place for something like a month. So you can see the upper layers getting drier earlier, while the deeper layers remain relatively with water over the time in the drought in the right side. Even when we irrigated, because we're working with sand, it does not have water, it's not holding water, so you can see the soil water content getting lower even when we irrigate the plants. So as for the root to shoot ratio, you can see here there's a difference between the cultivated and the wild watermelon. You can see in the cultivated, the ratio tends to the shoot, while in the wild watermelon, the ratio tends more to the root, which is not surprising. We know it well. That while the plants have more roots, the ratio tends to the root instead of the shoot. And even under drought, so it increases its ratio and go more. Actually, this is not statistically different. We didn't see any significant difference in the ratio between the ratio remained pretty much the same even under drought and under controlled conditions. When we look in the intrinsic water use efficiency, the ratio between the stomata conductance and the carbon assimilation, we didn't see any significant difference between the plant, which means they opening the stomata the same and assimilate carbon dioxide in the same way. There's no difference there. An interesting thing that we saw that with this measurement took with the LICO 600, the porometer. So you can see in the upper panels, you can see the stomata conductance, which didn't show any much of a difference between the plants. However, when we look in the lower panel, we see something more interesting when you're talking. This is the photosynthetic efficiency, how much energy goes to the photosystem to the creation of ATP and ADPH. And you can see that when we look at in the wild watermelon, you can see the trend is opposite to the domesticated watermelon. Over here, you can see when it drought, the values are reduced. However, we see the opposite in the wild watermelon. You can see when we have high soil water content, you can see a reduction in the photosynthetic efficiency, which means that, and in the opposite, under low soil water content, you can see it's pretty much stable, and which means that this plant is pretty much sensitive to logging or to high soil water content. When we did a blue section of the leaves, you can see this difference. We have no difference between the irrigated and the drought treatment here, but you can see a difference between the wild and the domesticated watermelon here. You can see very much thicker leaves, much thicker cuticula in the wild watermelon comparing to the domesticated watermelon, and which again it's all well known from other wild species. Nothing very interesting here. All our findings until this point is not something that you can take if you want to make a breeding and take traits from the wild into the domesticated. Is nothing interesting here. However, when we look at the root system, we get very interesting results. So you can see over here, this is the in the left we have the root weight on the different soil layers, and you can see here the actual difference between both domesticated and wild watermelon, and also between irrigated and drought. You can see that most of the biomass of the root system it's allocated in the upper soil layer. However, when we go to the root surface area, we have a different story. You can see that the domesticated watermelon over here, even under drought, the most of the surface is in the upper layer of the soil. But if you can see in the upper panel, this is the Citrus colocynthis, the wild watermelon. You can see that under control condition, it's pretty much the same as the cultivated watermelon. Most of the surface, root surface area is in the upper layer. But when you get it into drought, most of the root surface area are going to the deeper layers. And this is very interesting. You can see it over here in this picture. I don't know how much you can see it over here. This is the irrigated. You can see the root system is even a bit smaller, but there's no difference between the different depths. But in the drought, maybe it's a bit difficult to see here, but most of the root system, most of the fine roots are here in the deeper layers of the in the deeper layer. And when we scan the root system, this is in collaboration with Phenoroot, you can see that domesticated have a reduction in the root system biomass and size, and the colocynthis root system remain pretty much the same. You can see it over here in the chart, a reduction in the cultivated, but also pretty much the same in the wild watermelon. So, to summary, so colocynthis exhibit a high drought tolerance, but found to be sensitive to high soil water content. The drought resilience is primarily from the root system plasticity, which is not found in the domesticated watermelon. Without any additional carbon investment, you have much more efficient root system that get the water from the deeper soil layers. This trait offers strong potential for enhancing drought tolerance in watermelon with minimal impact on the yield. So our future plan, we are currently developing a population of the crossing between the wild and domesticated watermelon. This last summer, we tried in the field, we already found several accessions that have demonstrate a large potential. In the next spring, we do another experiment, also again in the tubes and also in the field to these specific accessions. So I wish to thank to my lab members, to the carbobite units in VAR, Chishti for the help with the seedlings, and to Bard for the funding. And thank you for listening.
Thank you, Amnon. Questions?
Hi, thank you for the talk. I was simply wondering if you foresee any drawbacks for doing the crossing. Like if you take a breed that is wild and you take a breed that is domesticated, there are usually a few drawbacks or a few concerns. Do you have any?
Actually, this fruit is horrible. By the way, used to, in the past, in the 19th century, it was been grown in the Gaza area, and been shipped to Europe to help with stomach issues, because it's causing to what do you call free activity in the stomach. Let's say it this way. So its taste is horrible. It's a small fruit, and so as we see it, the main potential is the rootstock, because watermelon is already grafted in Israel, mainly because soil-borne diseases. So we look at it as a potential for rootstock. But again, in the field, we only in the population is a very early stage. We're now in F3, so we now going to F4. And we have many problems because there's male sterility, there's many issues there, but the potential is there. I didn't put we have a picture of the accession that was very successful in the field, but you can see the field is over-dried. We didn't irrigate it from the end of April, and I took the picture in the end of July, and the plant was totally fine.
Maybe I got something wrong, but at the beginning you showed the bottom layer was the driest, if I'm not mistaken. At the beginning of the presentation, you showed that under the drought conditions, there was a very low water content at the bottom layer of the soil. No, maybe I.
Okay.
More questions? More people?
What happened with the flavors of the fruit? It changed?
Short answer?
Horrible.
Okay. Thank you, Amnon. Before we move to the last talk of today, I want to invite you all to come again today later at 5 in the afternoon. We will have another session of Dryland plant ecology, but more focused on interactions with the biotic, abiotic factors. So we finished today with Aviad Karmazin, also from ARO, but also from Hebrew University, working on olive trees, and we are ready.
Very much. Work? Nice. Last one before lunch? Great. My name is Aviad. I'm a master's student at the Hebrew University. I'm doing my research at the Volkan Institute with David Yalin and also under Jose Grunswig. Leading this research is actually Hiba Butaye, and we get a lot of help from Tamir as well. Funding this research is the Israeli Ministry of Environmental Protection, and today I'll be talking about the effects that mulching has on olive cultivation. But first, I'll tell you guys a little bit of background. So, the Mediterranean climate is becoming increasingly dry, and because of this, more and more trees are experiencing drought stress. And this is putting olive cultivation under risk because most of the world's olives are grown in the Mediterranean. If you look at the actual practices that farmers use in cultivation of olives, you'll see that most of them trim their trees, and this is because that trimming the trees encourages an increase of yield. So if you trim the trees for many reasons, the yield increases, and so you end up with these big piles of branches and organic matter in the plots. And the best way to get rid of this, it's basically a bunch of organic waste. The best way to get rid of this is by burning it. It's quick, it's easy, it's cheap, it's not intensive for manual labor, and it's very common throughout the entire Mediterranean to burn heaps of olive trimmings. And it's also very common here in Israel, specifically in the Arab community. And the funding for this research is actually given to, well, the question is, can we give an alternative, a sustainable alternative to burning olive trimmings and getting rid of the waste in a less risky and wasteful manner? And that's what this research, the purpose of this research is to do. And so if we're looking at what can we do with the actual olive trimmings that's not burning them, the solution that comes to mind is using it as a mulch. So literally chopping up the trimmings, grinding them up, and then applying them in the same plot and using it as mulch around the trees. This is a picture from our plot. You can really see the wood chips. It's mulch. No need to explain. So what does mulch actually do? What do we think it's going to do in the plots? How is it going to benefit the cultivation? The first thing we think about, obviously, is mulch is on the soil. It's going to affect soil temperature and soil moisture and increase the moisture and make the temperature more stable, as well as provide a way to maybe seep nutrients into the soil, as well as possibly even help with carbon sequestration. So we set about answering these questions by setting up our plot here in Israel with 3 different treatments. A control group, a group with 5 centimeters of mulch, and one with 15 centimeters. 15 is very common in the literature, and if you ask most people, that's what will be recommended, but we also set about checking a 5 centimeter option because we feel that this might be more fiscally responsible for Arab farmers here in Israel. It might be more applicable. And so we tested all those groups' treatments on these effects, the soil water and the tree performance. Our plot is in central Israel next to Netanya. Our plot is unirrigated, so it's completely dependent on rain-fed. It's completely rain-fed and no irrigation, and we set up the plot with our treatments throughout the plot. We have 7 repetitions. That might be a lot, but we did that because olives tend to be very variable throughout themselves, and so we wanted to get as many repetitions as we could. And you see these colors for the different groups, so blue for 15, green for 5, and red for the control group. That's going to be throughout the presentation. You want to keep track. And this is what it actually looks like. You have a layer of mulch around the tree, and you can see the different treatments. We applied them in June of 2024, and we've been measuring since. We've been measuring the tree as well as the soil. In the tree, we've been measuring stem water potential, stemmetal conductance, and the diameter at breast height, as well as we were taking leaf samples, but that data isn't available yet. And for the soil, we measured soil moisture and temperature with sensors at 10 centimeters below the ground, as well as soil samples, and we used a Mira to take soil CO2 exchange. But again, these data aren't yet ready for presentation. So, results from all these measurements. The first thing we're going to look at is the soil water content. And over here you can see on the left side of the y-axis, you see the actual volumetric water content. That's how much water is in the soil. On the right side is the daily rainfall, which is represented by these gray bars. And in the beginning of this graph, you see that the control treatment seems to be doing the best. It's higher than the rest of them. And then throughout the year, as the year goes on, this is February 2025, as the year goes on, the mulched groups actually catch up and surpass them, specifically during rain events. So they gain more water during the rain events, but then soon afterwards drop quickly and they match again. And so it's a short-lived gain, but it is a substantial gain. And then if you want to look at the temperature in the soil as well, the treatment with the control group treatment is less stable. You can see it's the coldest one during the winter, and it's the hottest one during the summer. And the mulch groups are the opposite. They're much more stable throughout the year. But if you want to look at the stability of the temperature during the day, we also looked at this is 10 centimeters underneath, that's where our sensors are, so that's where most of the roots would be. And so we want to look at the actual daily amplitude, so how much extreme temperatures are the plants being exposed to throughout the day. And so we took the measurements of the hottest measurement in the day, the coldest measurement of the day throughout the year. And what you can tell is that the control group is much more variable. It's exposed to a lot of extreme temperatures as opposed to the mulch groups, which have a much more stable temperature throughout the day. This is probably because that when you add mulch, you're basically pushing the roots deeper in the soil, and we know that the deeper roots are in the soil, the more stable the temperature is. So we talked about soil and how this is going to actually affect the plant. So our first graph here is the diameter at breast height, and you can see here as well the grain measurements. And diameter at breast height has a natural kind of cycle where it'll increase during the spring after the rainfall of the winter, and then decrease during the summer as the plants here kind of conserve their resources. And so that's very much expected to see this rise during the spring and this decline here. But the interesting thing to look at here is during these 3 months, May, June, July, whereas the control group begins to decline in its diameter at breast height from May to June. During these months, the mulch groups, the 15 centimeter one, we didn't have enough hydrometers for the 5 centimeter treatment group, stay stagnant and only begins to decline from June to July. So that's one less month of summer diameter decline. As well as this last point here, right around mid-September, late October, there was the first rain of the season, and the mulch group responded much more significantly than the control group. Looking at other tree responses, we looked at stem water potential. So here on the y-axis, you have negative numbers. The more negative megapascals, the more negative it is, the more pressure the tree is putting on itself to pull water up. So if it's lower, it's probably exposed to more drought stress. And so this seasonal variance is expected, but there's no difference among the groups. And the same with stemmental conductance. So how much water is leaving the leaf area per second. That's the unit. Also, there's seasonal variance. That's very much expected, but definitely no differences amongst the treatment groups. And after all, we want to look at yield. How many kilograms of olives per tree are we actually going to produce? And we looked at yield from 2025 and 2024. And 2024 was a much nicer year. They had a lot more yield. You can see the mean averages across the groups in the yellow boxes. They're like in the 50s. And then this year it was very low, and nobody passed 20. This is because also, one reason is because olive trees have a natural mast year. They'll have 1 year where they'll have a higher yield and 1 year where they'll have a naturally lower yield to manage their resources. And then another reason could be because decline in rainfall in this plot is completely dependent on rainfall. So, in summary, what we talked about today is that mulch succeeded in stabilizing the temperatures throughout the year and also throughout the day, and exposed the temperatures to less extreme temperatures. And it also increased soil moisture for the trees. Even if it is briefly, it did succeed in increasing soil moisture during rain. And there's a seasonal advantage in the tree diameter, so it gains one more month of not declining in tree diameter, which in a dry land condition is very important. Every month matters, especially in the summer months. So what should you take away from this is that mulching is still valuable as opposed to burning tree trimmings, especially if you want to utilize all the moisture that you get, especially if you are rain-fed and you have no irrigation. And also, if you want to keep your roots in an optimal temperature for growth, the mulch can keep the temperature at a much more stable level throughout the year, which is good for tree growth. The tree performance isn't quite. Is mostly unchanged. We didn't see any responses in stem water potential or in stemmental conductance, but this is also the first year of the research, so we hope to see more changes throughout. We expect to see it more in the next year. And even after a dry year with very little rain, the trees were able to produce some yield, which is interesting to see. And in the future, we want to be looking at other data that we haven't looked at yet, which is carbon and nitrogen, especially assimilation and mobilization in the soil, and seeing how minerals interact with the mulch and the tree. And also also, we have a new additional research site in Dir Chana, which hopefully will give us a new prospect on how different soils react to mulching. Thank you very much.
Thank you, Aviad. Questions about only mulching?
Thank you for the talk. Two questions. One, did you measure the soil evaporation? And second, did you measure the deeper soil layers, soil water content?
No to both.
Hi, thanks. Nice talk. Also, two questions. One is more a technical question. The mulch, is it just grinded dry material, or is it actually kind of pre-treated or some sort of decomposition happening already? And if so, it would make a difference. And the second question is, do you plan maybe also to look at more nutrient data, nutrient retention, and also the biological world of microbes, if it changes microbiomes and so on?
So first off, microbes aren't my deal, so I don't think I'm going to be looking at microbes. Maybe, I don't think so. But you're talking about the degradation of the mulch and what the mulch actually is, and it's the tree trimmings of the season. So farmers will trim live trees, leave them to dry, and then burn them. So the idea is recent prunings from olive groves. And they haven't been degraded much when we first applied them, but recently we've been looking at the plots again, and you can see that there's a lot of change. An interesting thing that I noticed that I didn't post up here is that the roots actually are starting to grow up into the mulch. So what we might do in the future is actually take the mulch and sample it and check the mulch itself for changes in minerals and comparing that to the soil and the leaf samples.
More questions? Did you try to add, for example, to make another layer, for example, compost, and then on that put the mulch, and then increase nutrients and increase the biodiversity around those trees?
So that's not included in this research project, but I know that my mentor is looking at composting as an additional thing to mulching in different fruit orchards, but not in this research.
And more question. Aviad, you mentioned the 5 centimeter treatment to be more financially available for the farmers. So it has a cost. The mulching has a cost. It's something that is hard for farmers to do.
So if you're looking at the volume, 15 centimeters is 3 times as much as 5 centimeters of mulching. There is a cost to it because you have to bring the mulch to every tree. You have to spread the mulch. This is manual labor, and it's time and it's cost, and it's also the mechanization, the actual chopping of the tree, and all this is money. And if you tell a farmer to put 15 centimeters of mulch, that might not be cost effective, and then they'll opt to, well, I'm not going to put 15 centimeters, I'll just burn it. But if you tell them, okay, you can put just a little bit of mulch, enough is 5 centimeters, and maybe that is enough to cause a change, and that's what we were looking into here. And I think 5 centimeters is enough to create some change, but not as much as 15.
So thank you to Aviad for presenting this work. I'm David, the supervisor that you mentioned. Just to add about the financial viability of it, that to make the 15 centimeters of material, we had to bring in a truck load of material, and we're now estimating to understand if what comes off in the prunings actually amounts to 15 centimeters or more like 5 centimeters, and it looks more like the 5 centimeters. So.
Thank you, David. And we have an option to skip lunch. Do you want to stay here? Or we can go. Thank you so much for participating.